I went to the ladies this morning and I saw this screaming face drawn on the board, shouting with the hair all freezed out, shouting help. So I really hope that you are not feeling like that today. This is the last day of the workshop. It's nice to see that you all made it here. So it is a great pleasure to introduce to you Professor Feng Z. Li from the University of Pennsylvania. He is a professor of statistical genetics and genomics, and his interests include genome-wide association studies, the analysis of the human microbiome, heart failure, EQTL data, and genomics of ovarian cancer. So he will be presenting to us today work that he has done on microbiome, metagenomics, and high-dimensional compositional data.
Great, thank you very much. Thank you for inviting me. So this, the title of my talk is also a review paper I'm finishing. I think it's due April 1st to appear next year, actually, in the annual review of statistics and application. So in this review paper, I try to write some statistical problems, statistical questions related to analyzing the microbiome metagenomic data. So, we heard lots of talks, wonderful talks, talk about the association between microbiome and soil microbiome, biofuel, and microbiome plant. And we all know that microbiome, in the last couple of years, there are so many important publications in Nature Science indicating the association between human microbiome and the diseases. Almost all the diseases, you can find a paper that showing some type of associations. Some are convincing, some are not that convincing. So, and now we have a tool to really study, to look at the human gut microbiome through a next generation sequencing. So we got all those data. We heard lots of talks talk about how to analyze the data, how to identify what are the microbes sit in the human gut, what are the compositions. So we are interested in understanding for example, how many of them, how they interact with each other, how the composition of the microbes respond to environmental and biological changes, and how the change of the composition are related to human health and the risk of diseases. So what I like to do today, I just want to show you some of the studies that I am involved in human microbiome studies, and talk about some of the statistical issues. So the first project I was involved is a study at University of Pennsylvania to look at, is a cross-sectional study to look at the association between diet and human stool microbiome composition. And this work was done in collaboration with two GI doctors, Gary Wu and Jim Lewis, and a microbiologist, Rick Bushman. So this was done a couple of years ago. What we did, we collected the data, the fecal sample of 98 healthy individuals, and make sure they were not on antibiotics. And we did a 16S sequencing. Eventually get out of this 98 individuals about 900,000 reads, the 16S RNA reads, and the number of reads varies from sample to sample. Some individual we only got 2,000 reads, some sample we got 16,000. The mean about 9,000 reads, and those 900,000 reads can be into OTUs at 97% levels, about 1,700 OTUs. And those OTUs can be further been into genera, 119 genera. So those are the microbiome data. In addition, we also have a questionnaire, frequency questionnaire, FFQ. And so basically it's a long questionnaire, and people use this a lot in nutrition study. And based on this questionnaire, you can score the micronutrient. There's a computer program, you can convert the information from the questionnaire to quantitative measurement, some quantitative measurement of micronutrient. We also have data on demographic data such as BMI. So the original goals of the study are twofold. One is to assess the association between the Euro diet and microbiome composition. And we also want to study the association between microbiome and BMI after adjusting for diet, for total caloric intake. We want to see whether there is still any association between microbiome and BMI. So those are the data. I'll just give you some idea about what the data look like. You saw this type of table very often. This is the OTU abundance table. So each row is one sample, each column is one OTU, and what we plot here is a log 2 of the count of the 16S reads count. I just want to point out, and here you can, based on the OTU reads, the sequence, you can also build a phylogenetic tree. And so I just want to point a few things. One is some of the OTUs, they are very abundant. You observe those OTUs in almost all the samples with a very high abundance. Some are very rare. Some OTUs only appear in a few samples. Similarly, for a given sample, you only observe, you don't observe all the OTUs, you observe some of the OTUs. So the data you have is this counts data. It's a sparse counts data, high dimension, sparse. Sparse means you have lots of zero, and the data are the counts. Those are the OTU tables. So initially what we did is this very simple correlation analysis. So for each of those micronutrients, remember those are the micronutrients, and this is about 20 relatively common bacterial genera, and we just calculate pairwise correlation just to have some idea about whether there's any correlation between the micronutrient and the abundance, relative abundance of those genera. And you can just see, so those are the Rank-Spielman correlation for each pair, for each given genus and each micronutrient. You can just calculate the correlation. But you can clearly see different type of correlations. For example, Bacteroides, you see a positive correlation with amino acid and choline, and negative correlation, for example, with carbohydrate. On the other hand, if you look at Prevotella, you see a positive correlation with carbohydrate, but negative correlation with choline. And this sort of a differential correlation, you can observe this. So this is very simple pairwise correlation analysis. You can do some pairwise tests, do FDR control sort of things. And just to confirm what we observed, we also fit a statistical model to the data. The first model we fit is a very typical standard Dirichlet multinomial regression, but it's a sparse version of the Dirichlet multinomial regression. So the question we want to ask, remember, with that data is which micronutrients are associated with which bacteria genera with a genus association. The data we have for the IC individual, we have Q bacteria genus counts. So those are the counts we observed. And we have P covariate, which are the micronutrient. In our case, P is pretty big. It's hundreds, 119 different micronutrient. No, this is roughly 119. This is like 200 micronutrient. We try to understand how they are related, how those micronutrients are related to the composition of the bacteria genera. So that's the goal. So the risk multinomial model has been studied in the context of microbiome data analysis. Chris Queens had an original paper talk about to use the mixture of the risk multinomial to model the distribution of the count in the context of microbiome data analysis. So the basic idea, the model is very simple. Remember, we have, for a given individual, we have total number of counts, and we distribute those counts into different bacteria genera. So you have the joint distribution of the count given the total follows a multinomial distribution with a parameter pi. So pi basically the true proportions for Q different bacteria genera. And in addition, you assume cross-individual, those pi follows the Richter distribution. So basically the true composition cross-individual follows the Richter distribution. And if you integrate out the pi, what you get are the marginal distribution, which is the Richter multinomial. And the reason you want to include this, the Richter component, is really try to control, to model what we call the over-dispersion. So over-dispersion, basically, the variability you observe in the data usually is much bigger than the variability that you can predict from the multinomial regression model. So this is basically a probability model to describe the distribution of the genus count for a given individual. And here the parameter in this model are the gammas, and the gamma parameter. And those gamma are related to the expected value of the count given the total. So gamma is proportional to the expected value of the count. And regression analysis, basically you try to understand whether there are any covariate that are related to those gamma. So you can build a regression model. You assume, for example, a linear link for the J's genus taxon. You can model the effect of the covariate on the mean of that relative mean, the mean of that genus. So P is a number of covariate. In our case, it's a measurement of micronutrient. So in this model, basically, you have a regression coefficient matrix, right, for each of the LTU or taxon. And so each one you have, so those each column represents one micronutrient. And the coefficient, for example, you look at the case micronutrient, this beta regression coefficient beta vector summarize the effect of the case nutrient on the distribution of the Q bacteria genera, basically, right? And for each of the given genus, those beta summarize how different nutrients affect the abundance of this particular OTU. So that's the interpretation of the beta. So this coefficient matrix, of course, is a high dimension. You have Q is the number of the bacteria taxa you are considering, and here is the number of micronutrient. It's a big regression coefficient matrix. So in order to estimate this matrix, we need to make some sparsity assumption. So the assumption we make is we assume out of this many nutrients, micronutrients we considered, not all of them are related to the distribution, the composition of the bacteria genera. And for a given bacteria genera, we assume not all the nutrients are relevant. For a given, if the nutrient is relevant, they may not be associated with the distribution of all the bacteria genera. So in a sense, we assume this matrix are sparse, are column-wise sparse, and overall is all sparse. And you can capture this assumption when you try to estimate this regression coefficient. So this is actually now is becoming a very standard way of estimating sparse regression model. It's through some regularization or penalization. So what we did is we proposed a penalized likelihood approach to estimate the regression parameter. And then there are two penalties. So this is negative log likelihood as our objective function. And there are two penalty functions. One is make sure the column are sparse. Another penalty function, which is the L1 penalty, make sure the overall matrix is sparse. So, and you can do this by very efficient algorithm, block-wise coordinate descent algorithm. You can fit the model, and in this model there are two key tuning parameters we have to tune. One is the lambda, the other is the C. Lambda determines how sparse the model is. Usually what people do is you can do a BIC or cross-validation to choose the tuning parameters. So this is what we got. So instead of look at all the 119 bacterial genera, we just look at relatively common ones, and that's the heat map I showed you, the 30 relatively common bacterial genera as a response, and we consider 119 micronutrient in the model. 119 nutrient, the total number of micronutrient is bigger than 119, but some of them are highly correlated because they all scored from the same questionnaire. So we look at 119. Eventually, the model I described select 11 nutrients, 11 micronutrients, and 13 genera to show some type of association. And so those are the 13 bacteria genera, those are the 11 micronutrients. I'll not go to the detail of this result, but just want to point out there's a couple of sugar, like maltose, sucrose, choline, all sugar-related micronutrients. There's animal protein, fat, somewhat related to the distribution, composition of those 13 bacteria genera. So you can also get this regression coefficient. The estimate of regression coefficient give you further detail about how they are related. For each column is the nutrients we selected, and the coefficient tell you, this particular nutrient is associated with the composition of which particular bacteria taxa. We can also obtain the p-value for each of the nutrient and the bootstrap selection probability. Bootstrap selection probability, basically, remember I have tuning parameters. Usually the results are sensitive to the tuning parameters. So usually what we do is run this model multiple times, 100 times by taking a bootstrap sample. And we count how many times out of this 100 run, how many times we select those nutrients. Most of them, the selection probability is high. Some of them is pretty low, like this. Out of 100 run, we only observe 35 times that this nutrient being selected. So this is probably a false positive. But in any case, I want to show you this model fits the data reasonably well. So this is the standard Dirichlet multinomial model fit to the count. This is square root of fitted count. This is observed count without adjusting for the micronutrient. So the model can capture the variability of the data reasonably well. And this is the one that we include some of the micronutrients in the model. So this definitely improve the fit of the data. So now R square go from 0.7 to 0.79. It changes the likelihood of 120. So I think that the nutrients certainly, micronutrients certainly explain more variability of the data that you observed. So this is basically, I just want to show you this model can be used to model the count, the joint distribution of the count. And this model can be used to test whether a particular covariate has any effect to the overall composition of the bacteria taxa. So next question we want to answer is, we want to find out which particular bacteria genera that are associated with body mass index. Remember, we also have the body mass index information. So here the idea is you have, for example, 100 bacteria genera. You want to find a subset of them, and they are subcomposition of the subset of genera may be related to the BMI. Now here's the issue I want to talk about is related to the compositional data. Remember, most of times when you run the program, for example, Megan or many other programs for the species quantification or bacterial quantification, what you get are the proportions, right? They give you a proportion, a composition. So that's what we call the compositional data. So for example, if you consider P genera or P species, POTU, whatever, usually the number you get from running those programs are the proportions, right? For the I subject, we have P taxa, and the X are the proportions. Means the sum of the X has to be 1 and the X has to be positive. In addition, for many of the microbiome studies, not only we have the proportions, we also have lots of zeros. For a given sample, for a given individual sample, if you consider P is the number of species, lots of time you have zeros, right? For a given sample, you have zeros. So the data we have is a high-dimensional data, it's a compositional data, and it's very sparse because you have lots of zeros. This can create some problems in downstream data analysis. So this is what we call the compositional data. Essentially, your data point, your X, sit in the simplex, in the p-1 dimensional simplex. So the data do not belong to a Euclidean space. So this is what we call the simplex data or compositional data. Now, zero is very difficult to deal with. I'm not going to talk too much about how to deal with this zero, but I want to just show you some result on how to model the compositional data and why this is relevant. Say, for example, if you want to do regression analysis, in our case, we have Y to be the BMI, body mass index, and our predictor, now the predictor are the composition of the P bacteria taxa, right? And they sit in the p-1 dimensional simplex. And we want to associate this bacterial composition to the outcome through a regression model. And how to do this? So naively, you can say, okay, I'm going to fit a standard linear, to take a linear combination of the P composition, the P component of the composition. This is not ideal. First of all, your regression coefficient, this matrix, design matrix, is a singular matrix because for each row, for each individual, sum of the X is 1. Secondly, what is the interpretation of beta K? Beta K interpretation in regression setting is you fix all the other X, you change one unit of XK and see how the mean will change. But if you have compositional data, you cannot only change XK without changing other X, right? Because of these constraints. So this makes the interpretation hard if you just naively fit a standard regression model using all the P component of your X vector. So this problem has been recognized in statistics literature, mainly by John Aitchison in UK in the 80s. And he wrote lots of papers in the 80s. In one paper in Biometrica, he proposed, instead of doing this, you take a log of the ratio, which really makes sense. He said, what really matters is the relative ratio of two components that are related to the Y, to the outcome. And take a relative ratio. This is a good idea, very good idea. Now you reduce the number of parameters, regression parameters, from P to p-1. That's how you get rid of these constraints. Now your log of the ratio, there's no constraints anymore, right? So you can do the standard regression analysis. The only problem is you have to pick one group XP, the one group as a baseline, in order to compute this log of the ratio. And your result can be different for different XP, particularly when P is large. So if you are interested in variable selection, you're interested in select which variables, which bacteria taxa are related to the outcome, the result may depend on what you put in the denominator. But if you expand his log ratio idea one further by introduce one additional regression parameter, beta P, now you're back to a P regression coefficient, but you have to put the constraints on the regression coefficient. The constraints are very simple. It's the sum of the beta has to be 0. Now the interpretation is easier. See, if you look at this, you do regression analysis under the constraints that the beta, sum of the beta being 0, now you will never select one variable. At least you have two, right? You cannot have just one non-zero beta to make this equality hold. So that's the idea. So now we propose a generalized linear regression model with a linear constraints on the regression coefficient. Now I don't have to choose a baseline group. In addition, what is nice about this model is invariant to the total number of taxa. Doesn't matter what is the n. For different n, because the sum of beta to be 0, this beta coefficient are invariant. It's that you can have like 3,000 counts, you can have 10,000 counts. The interpretation of the beta are the same. So this is invariant to the number of taxa. So this is how we deal with this composition nature of the covariate by putting a constraint on the regression coefficient. And so under this model, you can do variable selection. For example, we are interested in selecting a subset of bacterial genera that are related to the BMI. You can do regularization, do standard penalized regression, but you have to, this is so-called lasso regression, but you have to do this under the constraints that sum of the beta being 0. And this makes the algorithm a little bit more complicated because of these constraints, but that can be done. So back to the data, I showed you 98 individuals. We start with 87 bacteria genera. We want to see which one are related to body mass index. And this model, again, for all those regularized estimation procedure, there's this tuning parameter lambda you have to choose, and the result can depend on how you choose the lambda. So often we do bootstrap again. So those are the 4 bacteria genera that we selected based on our data, based on the bootstrap selecting probability. So this is just a naive, take a bootstrap sample, rerun everything, greater than 70%, we select 4 of them. We also calculate the formal stability selection probability. They're all pretty high, and this is the estimated coefficient. Those are the 4 bacteria genera that we selected in the model. Those are fairly stable. So this is the bootstrap, 100 bootstrap run. Those are the 4 bacteria genera that we selected. Now, this one is we just look at the observed BMI and the fitted BMI with those 4 bacteria genera to get this, give you some idea about how well the model can explain the variability of the BMI. This is R-squared 0.3. It's not huge, but I want to point out this. If you look at the literature, what people reported is that for the BMI, they look at how much the human host variance can explain the BMI variability. What they estimate is 17% of the variance of BMI that can be explained by considering all the SNPs, all the autosomal SNPs, can explain 17% of the variance of BMI. But in our data set, if this R-squared 0.3% is truly true, this is actually is very informative, right? Basically says 4 bacterial genera can explain more variability of BMI than you consider the genetic variance. But I want to point out there's a problem with this R-squared estimation. The problem is because of this regularization, this penalty function we put in there, that the estimate of beta are biased. Estimate beta biases lead to the biased estimate of R-squared. So that R-squared may not be a very reliable estimate. So just recently, we figured out a way to get a better estimate of the regression coefficient, so-called D-bias estimate. And this is really follows a tech report from Stanford group. They propose a way to correct the biasness to obtain the unbiased estimate of the regression coefficient. I'm not going to basically do some projection. In addition, you actually can get asymptotic expansion of this unbiased estimate of the beta, and you can obtain the asymptotic distribution. From that, you can obtain the confidence interval. And this is what I found is convenient. We use that procedure again. Those are the 95% confidence interval for all the bacteria genera, the beta, the estimate of the beta, exactly the same for bacteria genera that their 95% confidence interval not including 0. So I think if you look at the confidence interval, if you look at the selection probability, they all point to those 4 different bacteria genera that are associated with BMI. So use this unbiased estimate of beta, we can recalculate R-squared. Now this time the R-squared is 0.23, and this is how they fitted BMI and observed BMI, the plot. But still, 23% as compared to 17%, I think it's still important to know how much of the variance that can be explained by only 4 bacteria genera. Okay, so this is a regression problem. This is how we do regression analysis. So next question, the third question is, if you look at the literature, a lot of papers, you have this composition, right? You use Megan, use whatever program, you get composition, then people calculate the standard correlation, Pearson's correlation on those proportional data, and to try to understand the correlation structure of the relative abundance of the bacteria taxa. So I think considering the correlation makes sense because the correlation structure, co-occurrence correlation, give you some idea about the cooperative or competitive relationship among different bacteria taxa. But there's a problem. Ideally, we want to compute the correlation or correlation matrix based on the true count, right? So if you consider P taxa, we really want to know how many particular taxon this I carry. If we get a true count of bacteria taxa from each one of us, we can look at the correlation. But you never get this, right? We never get the true count. And so this is a random vector, p-dimensional random vector, which is unobserved. What we observed from the sequencing data, from the metagenomic data, are the proportions, so the estimate of the proportion, which you said you convert the true abundance into some estimate of the proportions. Now the thing is, you cannot compute the Pearson's correlation based on its proportions. It can be very dangerous. You can get the spurious correlations. And this actually has been recognized by Pearson. By Pearson in his very original paper. It's not a good idea to compute the correlation if your data sit in a simplex. And again, Aitchison in the 80s really noticed this problem. And most recently, Friedman and Alp in the PLOS computational biology paper also point out this issue. It's not always an issue, sometimes could be a big problem. So let me just show you what is the problem. So this is just a simple demonstration. If I simulate data, this is a true log of, the W is a true count of the bacterial taxa. You assume this W follows a log normal distribution and assume they are totally independent. You generate the W, you convert it into proportion. Then you calculate the Pearson's correlation based on the proportion. You can see a correlation of 0.6. So those are the box plot of the pairwise correlation. You shouldn't observe any correlations because they are independently generated. But if you calculate correlation, you see a bigger correlation there. And this really, this spurious correlation depend on the distribution, depend on the structure of the mean. The most severe case, if you have a few very abundant taxa and a few low abundant taxa, you will see a lot of spurious correlation. And the reason is very simple, is if there's a very abundant one, if you increase this abundance one, those less abundant one will decrease. They all decrease create the false positive correlation. And this is why you see a lot of false spurious positive correlations. So the point I want to make is you cannot sometimes, not always, sometimes you cannot just simply calculate the standard correlation based on the compositional data. It can be dangerous. So the question is there a way we can still estimate the covariance structure based on the compositional data? And there is a way to do this under some assumption. The assumption is if the covariance matrix is sparse, means if you don't expect all the bacterial taxa are correlated, then it's possible you can estimate this matrix based on this nice observation. So remember, xi, xj are the relative abundance. Those are the proportions for the i's taxon and j's taxon. You take a log of the ratio. Those are, and you take the variance. You can estimate this quantity, right, based on observation, because we observe xi and xj. Then you take a log of the ratio, take the variance. Now the variance or log of the ratio is the same as the variance or log of the true abundance, the true counts. They are the same, right, because there's a constant here. And if you expand this, basically the variance of log of the ratio can be written as the variance, the true variance of log of the count minus the covariance matrix, which is the one we want to estimate. So you can write this into a matrix form, and this is a variance matrix of all the pairwise log of the ratio can be written as a vector, basically can written as a two matrix plus another matrix. And those two matrices are the low rank matrix. Those two together is rank 2 matrix. And this is a matrix of interest we want to estimate. And if you assume this is a sparse, remember this is what we can estimate from the data. This is a rank 2 matrix. If this covariance matrix is sparse, there's a way you can estimate them. And there's a lot of literature on this. You decompose observed matrix into low rank matrix and sparse matrix. And we have a procedure of doing this. The idea is very simple. You project your matrix into a rank 2 matrix because this matrix is sparse. Take a residual, do the thresholding, you can get an estimate. And there's a very strong theoretical support for this procedure. So let me show you what we can get. Again, this is a simulated data. We simulate, for example, 50 genera, sample size 100. What I showed you here is ROC plot. So this is, remember, our goal is to recover the sparsity pattern of the covariance matrix. And what I show here is a false positive and true positive rate for, let's just show you. This one line is if you truly observe the count, then you can just use a count to compute the covariance matrix. And this is this line. And there's another line is the procedure based on the sparse decomposition procedure I described. This is the one if you just calculate the correlation based on the proportion data, and you can have a lot of false positive result from that if you just simply compute the Pearson's correlation and do some thresholding. So we did a lot of evaluation of this procedure. It's always performed extremely well, especially when the covariance matrix is sparse. The sparser the covariance matrix, the better the performance. That's what we should expect. So I think there is a hope to compute the covariance matrix based on compositional data if indeed the matrix is sparse. I'll skip this. And this is what we can get. I don't know, I need some help to interpret this, but anyway, so we apply this procedure to our data with a 52 genera, and this is the covariance matrix. This basically tell you which bacteria genus is associated with another one, and I have positive association, negative association. It's very difficult to interpret this because a lot of genera we don't know anything about it. But this, nevertheless, we do get this sparse estimate of the covariance matrix there. So now so this is some work we have done related to compositional data. And I think some care need to be taken when you use this type of compositional data. You cannot just use, even for the principal component analysis, you cannot do standard principal component analysis on the compositional data directly. You need to do something. Next, I want to just talk a little bit about another study, which is an ongoing study. We're still analyzing the data. It's related to the treatment of Crohn's disease. So this is a pediatric Crohn's disease. We have 90 children with Crohn's disease, and they were treated by two different treatments. One is alimental diet treatment. The other is anti-TNF therapy. So those are the two standard treatments for Crohn's disease. What we got is at the baseline level, we have diet information, stool microbiome, and we did a shotgun metagenomics. At the baseline, week 1, 4, and 8. So this is a longitudinal follow-up study. And at each time point, we assess whether the children respond to the treatment or not, as measured by two clinical measurements. One is PCDAI, the other is FCP. We have this information. So total, we have 90 times 4 shotgun metagenomic data. I just want to show you, you probably all know this. So this is we use the metaflam to quantify the species abundance. So I just want to show you the plot, how many reads, the number of reads we can map to the metaflam markers. They varies a lot from sample to sample. You know, some sample get very small number of reads we can map to those markers. One of the reasons is some of the kids, we actually observe lots of human DNAs in their sample. And recently we figured out why for some individuals we have lots of human DNAs because they stool sample their blood. From the blood you get the human DNAs. That's why for some of the samples, the number of reads we can map to the metaflam marker is small. So we heard there's a lot of procedures, lots of algorithms now available to quantify the relative abundance of the bacteria taxa, and many of you are the developer of those programs. We use a MetaFlan. The reason is it's fast, and so we use a MetaFlan. And I want to point out, I think this is true for all those procedures. They do one sample at a time. They do analysis, right? Look at one sample, give me a quantification, run another sample. So I want to show you an approach based on multi-sample approach. And the reason I want to do multi-sample means I want to quantify the relative abundance by look at the data from multiple samples, from all the samples together. The reason for doing this, one is I want to account for marker-specific effect I'll show you. The other is I can provide a variance estimate, right? You get a quantification, usually you like to know the variability. So you want to have a variance estimate. In addition, this joint analysis actually also help us to perform downstream analysis, such as differential abundance analysis or other regression analysis. So this is what we observed, so-called marker-specific effect. So this is a sample, right? Each row is a sample. Each column is a marker. So this is for E. coli. Those are the MetaFlan markers. What I plot here are the normalized by the length of the marker, the number of renormalized length of the marker. Normalized count for each of the markers. And you can clearly see this marker-specific effect. So in theory, if the markers are perfect for this E. coli, you should roughly observe similar number of counts after you adjust for the length of the marker, right? Cross marker for each sample. But you can clearly see some markers you have more counts than another markers. And this is for E. coli, this is for another species. You see a very clear pattern. And this pattern are fairly consistent across multiple samples. So this is what I'm saying. You cannot simply just add the count over all those markers. So those marker quality are different. You want to account for that. And the only way you can account for that is to look at the data across multiple samples, right? To multiple samples, you see some consistent pattern, and you try to adjust for it. And this actually can be done by fitting a very simple model. You consider one species. The data you observe for the IC individual, the case marker gene, and you have a count. And you assume this count follows a Poisson distribution with a parameter theta I is the abundance index for the I sample, theta I here. And of course, this also depends on the total number of the reads which can map to the markers, the Li. Mk is the length of that particular marker. But I have this parameter phi k. For the case marker, this is what we call the marker-specific effect to account for the variability of the data you observe from marker to marker. So you have this phi k, and so basically what you observe not only depend on the true abundance of the IC individual, also depend on which marker you're talking about. And you have to make some assumption, for example, you put a constraint on phi k because you want to identify the parameters. So what MetaFlan does is essentially assuming this phi k to be constant. To be constant, you sum them together. Okay. So this simple model, and you can estimate this model. You can iteratively update this model. So let me just show you some results, simulation results. So this we simulate, for example, a simple population with 20 species. One species 62%, the other 19 species, their relative abundance 2%. This is what you can get from MetaFlan. And these are the number of reads. There's a lot of reads, a fewer number of reads. Sometimes you can get a very bad estimate. But if you fit the model, so basically we generate the data, we use their marker. This is a Poisson model. What the Poisson model can give you? Can get much better estimate, especially when the number of reads are not too many. So we also get bias estimate towards the end, but at the beginning, the estimate are fairly good. For low diversity case, high diversity case, this is our estimate. This is the MetaFlan estimate. For intermediate diversity case, our estimate, MetaFlan estimate. So I think that the marker-specific effect need to be accounted somewhat. That's why I asked you yesterday for other programs whether there's potentially such effect that we need to consider. You may not be a good idea just add all those reads together to get a quantity. I think the time ran out. Okay. So let me just talk about this 1 minute. Within this modeling framework, you don't have to quantify the relative abundance if your goal is to identify which bacteria taxa that have different abundance between two different conditions. You can very easily just model this data, right? This is a relative abundance index. If you have regression, if you have other covariate, you just do a statistical model. Then you can test whether there's a covariate effect on the true abundance based on the data you observed. You don't have to do this. I mean, Susan mentioned this. You don't have to do this multi-step way. You do this in a unified way, then you can account for all the variabilities you observe in the data. And this can be done, and we have a paper on this actually. I'm not going to show you this. So let me just add by thank my students, postdocs, collaborators, and thank you very much.
Is there any question?
So first of all, a wonderful talk. Thanks.
Thank you.
One of the things that I thought about looking at your talk and thinking about other things earlier is this estimate that we're trying to do of the distribution of taxa in a sample, right?
Yeah.
Which parts of that distribution are the most important clinically? How important is it to estimate the highly abundant taxa compared to the ones that are less abundant?
Yeah, that's very, very good question. See, in our analysis so far, we only focus on relative abundance
right.
taxa.
But it could be that a lot of what's driving the error
Different.
estimate is how well you estimate the most abundant taxa. In other words, the way people
I see.
measure error,
How to summarize them?
right?
Yeah.
The measurement of error
Yeah.
is this, the difference between the two distributions.
Yeah,
The
yeah.
distribution, the estimated distribution,
I
that's
see.
dominated by the error in the
Yeah.
most abundant taxon.
Yeah, that's why I don't like that quantity. I like just if you compare them, you just plot them which one is close to the truth instead of summarize this into a single number. But this one, I think you can see the potential biased estimate you can get from Metaflan. Right. So this is their estimate. This is our estimate. This is the truth. I do have a number, some error calculated based on this. I see what you mean, but I think you just plot them. Plot if you do simulation, you have true ones, you have your estimated, and see how close they are.
I think you're not understanding my question. I'll ask you later.
Okay. Yeah.
Yeah, I think it's very sensible to correct the read counts. I just wondered, do you need for this to assume that all the samples are the same, basically, so you have replicates and?
No. So say each sample they have their own abundance. So theta I is their abundance index, and what you observe, the key is what you observe cross markers will depend on some marker specific effect, right? So see this xik depend on the true abundance, also depend on which marker you're talking about. So it's those two parameters determine what you observed.
Abundance is sample specific.
It's a sample specific, and in order to estimate this 5K, you have to have multiple sample. Yeah, and I try to show you this pattern is indeed quite consistent cross sample. Otherwise, there's no way you can correct them, right? It's something systematic cross multiple sample, and you can try to correct them. Yeah.
So you compared the 17% of variance in BMI compared
The genome.
with 23%. I do want to point out that we're pretty sure that the causality in the genome case goes from having the SNPs to BMI. But in the causality in the case of microbiome is going to go.
I'm not saying anything related to causality. It's just R square, the percentage of variance which can be explained by by the combination of those four bacteria.
But you're just kind of saying I'm a statistician, so I don't have to care about this. I don't think it's not really The power of genetics is that the causality is pretty clear. Okay, but
in microbiome.
It is it's different.
That's we didn't make a big deal of that R square. Initially, I thought 30% versus 17%. That's huge.
Well,
Then
the SNPs in the human genome might relate to the microbiome, mightn't they?
Oh yeah.
In that
That
might
as
drive.
well, but it's just the point that it's not to compare those two numbers as if they somehow
now
relate to each other. They don't.
Basically saying Metagenomics is better than human genomics, is what you're saying.
That's what he was.
Oh, I'm saying it may explain more variability than the host genetic variant,
But you use the word explain.
right?
Okay, there is another question there.
Well, it's just a point. So I love the marker-specific biases, and I think the origin would probably be differential genome coverage within the metagenome. So if we take metagenome data sets, map them back onto individual reference genomes, within a genome, you
Yeah.
see variations of coverage, which I suspect would be consistent across samples. So what you're seeing there is that, say, a particular marker gene being used by Metaplant is getting proportionally more reads than another marker.
Yeah,
That's
exactly.
due to GC content,
Yeah,
its position in the genome respect
Yeah.
to the origin of replication.
yes.
Yeah.
Yes, that's a good. We actually plot this marker-specific effect estimate phi against the GC content of the marker.
And you found a relationship.
But it's not. It varies a lot from sample to sample. It's not a clear pattern. So we are thinking about whether we can use the GC contact to adjust for that.
Right.
It's a little difficult. It's no consistent pattern.
Yeah. I mean, there is just a lot of variation in coverage within the genome.
Yeah.
Perhaps not.
So we just use that single parameter phi to try to capture some of the variabilities.
So concerning the application of correlation coefficient to the
Yeah.
relative abundance data, I think the people who are doing this type of networking, they usually do not consider one correlation coefficient. So for example, they look at multiple similarity measures, like Curtis-Hattenhauer,
Yeah.
and then they just draw that network. So I think they do know that to some degree at least.
Yes.
And another question, when you've been showing this regression model in the beginning, so is that a good idea to look at some kind of a group lasso type of a technique there? Because it could be that there are multiple species, right, that are highly correlated to some particular, whatever, output BMI. And if you would really enforce sparsity like with L1, what you're doing, you're going to miss these co-correlated species. Am I right?
You co-correlated, yes, you are right, with this L1 penalty term in there.
So
Yeah,
something like elastic network, for example.
possible. Yes, that's possible. But again, you still need to do that under the constraints that some of the data being 0. That's the point I want to make. You cannot just do the standard lasso, standard elastic net without considering the constraints on the regression coefficient data. Yes.
So when you look at the diversity, and if you move upstream and you look for sources of error, so if you go into your BMI study, there are over 100 species of Clostridia. So when you're identifying and you're naming, there are over 100 species of Clostridia. There's one species of Allostipes. There are one of Allocinella, two
Yeah.
of Allostipes,
Yeah.
and there's one of the, it's 9 of Allostipes and
Clostridium.
Caucaceae, or Cynomenococcus, there's 2.
Yeah.
And so what I'm interested in is what happens when you use some kind of a system and you have a target that's 100 times larger than the others in terms of the accuracy of the methodology. Because it seems to me that the information content that you have in naming things or identifying things causes a lot of error that's very difficult to essentially weed out. And it trickles into your system downstream. How do you handle that?
I don't understand it.
So the thing is, when you say Clostridium, it's not
Yeah.
it's not a uniform group. When you say Allocinella, it becomes very uniform. So the probability when you do matching, say using 16S,
I see.
it's very easy to get a Clostridium because it's so dominant. And there's so many different species of Clostridium. And so how does this impact all of the downstream analysis when you put this information into your sparse matrix and then you try and determine which methods work the best? Because it seems to me that you're not working with uniform size targets.
That's true. I need to think about it. I don't have a good answer to that. Yeah.
So that Clostridium is just not one OTU of particular Clostridium. It's a
That mix of different things.
Hundreds.
oh
Mix of different things. I think.
Oh.
Yeah.
Okay. Sorry, I think I will stop it here and leave further discussion to the coffee break because we are running 10 minutes late. So coffee will be served outside, and can we meet again at 11 to continue with the talk? Thank you very much.
Thank you.